Okay. So these topics are going to be a little different from what we've been doing up to this point. So up to this point, we've really been thinking about the probability of genetics, or how things are directly inherited, or how we can see phenotypes and connect those back to the genes that they come from. And today we're going to talk more about techniques that are actually used both in research settings and in medical fields to try and get at a lot of the information that we have been using this semester. And your textbook and I have referenced experiments that people have done at various points. And you've also had some activities to introduce those to you if you've done some of them. But I'm going to assume that everybody did that, like the PCR activity at the way back beginning. And so we're also going to talk about some more detail about how to do that, how that works, and why that's the foundation of a lot of things too. So today is more about, okay, that's great. Now that we know things are inherited, we know this is kind of how it works. What do we do with that? How do we get to something usable? And that really focuses on this new field of molecular biology and molecular genetics, as well as the biotech sector, and specifically, what types of things can we get from this? And this is going to come with a lot of other questions, like, should we do that? And I think you're going to find that the answer to this is very gray, because on the one hand, we have a lot of power to determine what is happening and also to find answers to things, but those answers could be used in ways that we might not be comfortable with, and they create a lot of privacy issues. So we're going to talk a lot today about this idea of molecular biology and biotech, and also how they connect to human identity. So how can that be both a very positive thing for us and somewhat scary thing for us? It's important to remember that the field of genetics began a long, long time ago with things like Mendel, but molecular biology techniques are really not that old. And a lot of these have really opened up the ideas as to why. Why do we see what we see? Why do these things happen? When Mendel is looking at his genes, all he can say is, well, this seems to be passed from one individual to another, and it seems to give this phenotype, but why? And we've kind of tried to get at this a little bit too with the OMIM project. You had to pick a specific gene that was identified for a disorder and a specific protein from that gene and look at how it's affected. All of those databases that you've been using have all come out in the past 10 to 20 years as far as accessibility to the public. And all of these also rely on these key molecular genetic techniques. So they're all built on things like types of recombinant DNA technology, the polymerase chain reaction, the ability to now sequence things when we want to know what their genomes look like, and also the ability to make changes to the genome whenever we would like to. And this all kind of goes around this idea of genetic engineering, which is kind of a loaded term nowadays, kind of like this idea of GMOs is considered a loaded term. But all of these things together allow us to then answer those questions and connect things like phenotype to genotype. And the reason that they let us do that is because they allow us to work at the molecular level. So instead of just saying, well, we know this, it's inherited, we can say, where is it coming from? What is it making? What does that thing do? Where does it go? And we can try and piece together everything that's happening from there. There are a lot of major technologies that are used for all of the things that you might read about, and most of them are based on a handful of things. The biggest one is polymerase chain reaction. So PCR is probably the biggest DNA technology that is used, and it is the basis of almost all of these other technologies as well. The reason for that is it allows you to target specific places in the DNA of an organism or its RNA It lets you modify those things however you might like, and that allows you to do things like clone or recombinere. So PCR is really the basis of transgenics, but it's also the basis of diagnostic. It's the basis of all kinds of things. So this is really a huge technique that really opened up all of these possibilities. Of course, PCR cannot usually be used by itself. A lot of the time it's going to be used with gel electrophoresis. And these are probably the two things that you're most familiar about for these DNA technologies, although we will discuss them quite a bit today. And both of these together introduce the idea of DNA fingerprinting, which is about looking at specific differences between individuals. How will biotech topics manifest? It really is going to depend. Yes, you will, Sarah. It's really going to depend on the topic specifically. So we are going to do a little bit of practice today with PCR. So you would, for example, need to be able to design a primer or identify how large a primer product might be. Those are things that you may have to do at some point. So we will practice that a little bit. Some of them will be things like, how does this technique work? Or why would we want to use this? Or if we wanted to look at this, which of these might we pick? Stuff like that. But it's going to depend. So for example, you should understand how things like gel electrophoresis work. So if I were to show you a gel, you would understand what you were looking at. You should understand how PCR works. So why you would want to use this. You should also understand how to design a primer and how to determine how large a piece of PCR reaction will be. When we talk about DNA fingerprinting, there are things about how this is used, and you may want to think about implications for that or what you might use it for. We'll also talk about the value of sequencing over things like DNA fingerprinting. So sequencing and DNA fingerprinting have different benefits, even though they look at similar things. You're going to need to use, CRISPR in your presentation. So this won't be on your exam specifically, but it will be part of your presentation. Things like that. You may also have some questions about the ethics of doing these things. That doesn't mean that you should say what I think. It means that I want you to talk about what you think and why. All right, so stuff like that might be important as well. Now, as far as like how much of this part of this project or this reagent we add, I don't care. All right, we're looking at why we would do this, the basics of how we would do this, and what we get from that. I think it will be a little more clear as we talk about each of them, how much information you might need to know. All right. So this is probably, I'll very quickly mention these guys, sequencing and microarray today. Stop that. So these will be quick mentions. And then we probably will get to more of this at the beginning of next week, although I will post videos for you of this. And then we'll probably do a little more stuff talking about the use of GMOs and transgenics. So how we use these in our society. But also how we use things like this in medicine. Okay, so this will be next week's topic for the most part. And this week's topic is going to be setting up these basic things, which are the basis of pretty much all other molecular techniques. The other thing that has become a really, really long way in the past 10 years is the ability to see these things in both real time and the physical locations in which they exist. So for the most part, when you think of PCR, you're thinking of something that is in a tube or something that's inside a gel. But the reality is that PCR opens up things like this. So one of the things that we're able to do now that we could not do before is we can actually look at where within an organism something is located. So for example, we can use antibodies to stain or look for things where proteins are present. We can use special RNA probes to look at where RNA and DNA is located. We can also cause organisms to express genes where we want them to, so that we can see where those genes should be. Oops, why do you keep doing that? So for example, in this image here, where we're looking at, where we're looking at multiple colors, each of these colors represents a different level of expression for insulin. So remember, insulin is an important signaling molecule that's used to control blood sugar. And this is a fish embryo. This is a zebrafish. It's like a little alien. And what you're seeing here is this green area is the fish probe. So fish is going to detect RNAs. So this is expression. This is where insulin is expressed. And antibodies detect protein. So this is where the protein is. So you can see here that the insulin is made in the cells up along here, but that the protein itself is largely in the circulatory system. And we can see those things at the same time. And when we're looking inside specific cells, so here we see lots of different cells that are located. If we zoom in, you can see there's blue and green areas. Some of these cells, only these ones, are the ones that are making the insulin. So if we want to answer questions like, where is this protein going to be made? Or where is this gene expressed? We have a lot of ways to look at that. We can look at it from, a PCR-based method, or we can look at it from a visual method. And this gives us a lot of power. We can do this for our patients too. So you can do antibody staining and fish in tissues that are taken from individuals. And you can use that to identify where cells might be making something that you need them to make, or if they're not. So there are lots of ways that we can use these new technologies to try and get at what is happening. This connects to this idea of forward versus reverse genetics. There are two ways to think about solving genetic problems from a molecular standpoint. In forward genetics, you're going to be starting with a phenotype. And in reverse genetics, you're going to be starting with a gene. In both cases, the goal is to connect the phenotype to the gene. A good example of thinking about forward genetic approaches is that you might have a patient that has a phenotype, but you don't know why. So you need to come up with ways to identify where the phenotype is coming from. This allows you to go from phenotype to gene, you hope. In reverse genetics instead, you start with a gene or a gene region, and you want to know what that gene does. You may or may not have a specific phenotype in mind when you start. So each of these different approaches are valuable, but they're valuable for different things. If you want to figure out why a person has a disorder, a forward approach might be helpful to you. But if you want to figure out what that gene actually does once you find it, a reverse approach might work better. So a lot of times when we're trying to find places to start looking, we're going to start by looking at random mutations, or essentially the phenotypes that walk in off the street. Once we have that, and it tells us where to go, i.e. this gene, we can then say, okay, how do we get from that gene back to the phenotype? So forward genetics is using a phenotype to get to a gene, and reverse genetics is using a gene to get to a phenotype. And both of these are valuable for different reasons. They also use different techniques, although sometimes the techniques overlap, the reason for using them is different still. Are there any questions about the difference between forward and reverse genetics? You're doing both of these things in your OMIM project. For example, you started with a disease that you wanted to be able to find information about, and you asked, what is the gene about that? You then find the gene and say, why does this gene cause this problem? What does this mutation do specifically that could lead to the phenotype that I see? So you're actually doing both of these in your project. We're going to start by thinking about some of the different techniques that involve DNA. So for example, primarily today we'll talk about PCR, gel electrophoresis, and DNA fingerprinting, although that also will include a little bit of information about sequencing and microarrays. And then next week we'll talk about actually manipulating DNA and looking for phenotypes, things like cloning, transgenics, CRISPR, RNAi, and GWAS studies. While we're all familiar with PCR, at least we've heard of it before, what you might not know is that PCR is actually very variable. In fact, there are almost 40 different types of PCR that are used depending on what you want to accomplish. Some of these are used to make changes permanently to DNA. Some are used to specifically get DNA pieces. Some of them are used for large pieces of DNA, and some are used for really small ones. And each one is done a little differently. You don't have to keep them all straight, but I want you to know that just because it might seem like a very straightforward reaction doesn't mean that it can't be used for a lot of different purposes. So how does it work? Remember that DNA replication requires a lot of different enzymes, and that each of these enzymes performs a specific job. For example, the DNA is tightly wound before it can be replicated. A specific protein called gyrase is used to unwind the DNA, and helicase is used to separate the DNA to make it accessible to the polymerase. In order for replication to begin, a DNA primer has to be used. This gives a new place for DNA polymerase to begin making a new copy of the DNA. This happens because DNA polymerase can only add to one side of the growing chain. Remember that DNA is anti-parallel. This means that the new growth will go in the opposite direction, toward the 3' end. So this is moving toward this direction, and this one is moving in the opposite direction. In order to do this in a test tube, we need to have access to certain things. We need something to separate and unwind the DNA. We need something to make a primer or somewhere for the polymerase to start. We need a polymerase itself so that we can make a copy of the DNA that we're looking at. We also need dNTPs. If we don't have free bases like A, T, C, and G, we can't make a new strand, so we need to provide those too. We also need to be able to provide the ability to do this multiple times. The reason for that is because DNA is extremely small. In order to see the DNA, you need at least 5 nanograms. Generally, you want to use at least 10 to make sure you can see it at all. For example here, we want to be able to tell where the PCR product we're making is going to be. These are examples of PCR products. That means that each of these bands has to be greater than 5 nanograms for us to be able to see it at all. The average weight of a base pair is extremely small. It's about 650 Daltons. So that means that the weight of double-stranded DNA is the number of base pairs times the number of Daltons, or super tiny. Do you have to remember how to calculate this? No, but what I want you to understand is that you need a lot. The weight of the entire genome is 3.59 picograms. That means that one copy of your genome is not even enough to see it at all. In fact, to get to enough to load a gel, you would need almost 3,000 copies of your entire genome. So you can see kind of where I'm going with this. If you want to look at a specific gene, well, that's even smaller. So the purpose of PCR really is to make enough copies that you could see the DNA at all. So what if we only want to see a small piece? We don't have 3,000 copies, but we can make them. To do that, we need to know the sequence of the gene we want to look at, or we need to at least have some idea of what that might be. Remember that a gene is a unit of heredity. More accurately, though, it's a distinct sequence of nucleotides, which encodes the protein. The average human gene is actually pretty big. It's about 28,000 base pairs long. This means that the average gene weighs, well, even less. In fact, we would need more than 329 million copies of the average gene to be able to see it. That's a whole lot. And of course, how would we get a single gene? If all we did was make a ton of copies of the genome and cut them up, they might look like this. We don't want to have all these bands. Oops. We want to have a specific one. So we want to go from not having enough copies to having enough copies of exactly what we want to see. PCR lets us do this by allowing us to target specific regions of DNA and exponentially amplifying that specific area. We can then use that amplified area to do a lot of other things. For example, we can use the amplification to create clones for sequencing. We can use it to analyze whether or not a gene is present. We can use it to diagnose hereditary diseases, identify genetic fingerprints for paternity testing or forensics, and also to diagnose infectious diseases. This was invented quite a long while ago and was patented in 1985. So even though this seems like it's been around forever, it's actually only 37 years old. They received the Nobel Prize for PCR in 1993 because it is the foundation for so many other things. Similar to what we discussed before about what is needed for replication, PCR also has similar requirements. You need to start with DNA template. DNA doesn't come from thin air. We need something to use as a base. We need a primer, something specific to where we want the DNA to be made. We need DNTPs or free bases, some kind of polymerase that can make and read the DNA, and something to keep it at a pretty similar place that it would be in the body so that the enzyme's able to do its job. So all of the things that are clear from thinking about how DNA replicates on its own are also needed in order to make PCR happen. Essentially, PCR is in vitro or outside of the body DNA replication. Originally, all of these things were essentially gotten from bacteria. They would raise tons and tons and tons of bacteria, and they would take out the bacteria's polymerase and use that to try and do this. We don't really do that anymore. So where do we get these from? Primers are usually man-made. And the important thing here is that they have to be DNA primers, not RNA primers. Remember that RNA is transient. That means that it is not something that's going to last very long. So when we make primers, we want to use DNA primers because we want them to stay part of the product. RNA primers get removed. We don't want that. We want the DNA primers to become part of the new product. So let's say, for example, we're looking at these strands. We want to make a copy of the entire gene. We have our start over here and our stop over here. We want to make copies of both sides. Well, We need two primers, a forward primer and a reverse primer. And we want the primers to be able to attach to the opposite strand. When you see a primer sequence, it's important to remember that it's always written 5 to 3 prime. This means that the reverse primer will need to be flipped when you write it down. So what does that look like? Let's say, for example, this is the region that we want to be able to replicate. And here are our two primers. The forward primer is easy. It's already in the 5' 3' direction. But the reverse primer is backwards, 3' to 5', because that's the direction that this piece of DNA will be built. That means when we write that primer, we write it in reverse. T, C, A, and so on. So if you're given a set of primers, remember that the reverse primer is going to be backwards from where it needs to be in order to amplify this region. When the primers are made, we then add them to the PCR reaction. DNA polymerase can then add to the 3' end of the existing DNA primer. And this allows us to create a replicate. So we're going to start with double-stranded DNA. We want to be able to separate the DNA strands, but we don't want to include too many proteins because trying to include a protein to actually do everything is going to be really hard. Thankfully, hydrogen bonds are weak. This means that we can break them by raising the temperature. So the first step is to raise the temperature the DNA is in so that the strands separate from each other. We can then lower the temperature again, and this will cause some of the DNA to anneal or connect to the primers instead of the complementary DNA. This happens because new hydrogen bonds are able to form at the lower temperature between the primer and the base template. This is why it's important to make sure that the primers that you're using are going to be able to anneal at this temperature. Those of you who did the PCR activity know that the temperature at which the primers melt is an important factor for how well they'll work. Once the primer has annealed, you can then extend the stage. So at this point, you want to keep the PCR products around body temperature or room temperature. This is pretty warm though, 72. Body temperature would be 35. So what we're doing here is we're increasing speed. It's not so hot that we're going to have the hydrogen bonds break. And it's not so cold that the hydrogen bonds will become more permanent or take forever. This allows the polymerase to function at its optimal temperature. Because the polymerase comes from bacteria that are used to being at high temperatures, we can get away with this. If you were to use DNA polymerase from your own body, this would be too hot. Essentially, what we're going to do then is we're going to repeat this over and over and over again. Every time we repeat it, we'll get more copies. And we need millions of copies. So that's what we really want to do. We start with our initial template. We heat the template to denature or separate the strands. We add our primers so that they can connect to them, and then we extend. And we do this over and over and over again until we accumulate enough products that we can see them on a gel. Normally, this is going to take somewhere between 25 and 35 cycles to be able to see. And these are lovely. But why are they all the same size? Well, this has to do with what is happening. So here's our initial DNA. And we added our primers. When we make these new pieces of DNA, they're going to build all the way past the primer on the bottom strand and all the way past the primer on the top strand. That's one cycle. In the next cycle, we have multiple templates now, not just two. We have one that looks like this, one that looks like this, and the two original templates. When they go through the denaturation process again, we'll have new primers added. What do you notice about the blue templates in comparison to the original templates? This one here is going to elongate in this way. This one will elongate here. This one will elongate here. And this one will elongate here. What's going to happen to the blue templates that didn't happen to the red ones? How does the PCR know when to stop replicating? No ideas at all. How does DNA replication usually know when to stop? That's right, Sarah, when it runs out of bases to complement. Stop codons are used for translation, not for DNA replication, but they are useful for stopping that. So here's the problem. These products have an endpoint that the previous template does not have. This means that these pieces can only elongate until the endpoint. The location of the previous primer is reached. Whereas these will continue replicating the entire piece. This means that over time, the fragments that are exactly the distance between the two primers will increase at a greater rate. In the next round, we'll have some that are exactly the size of the product, some that are a little bit too long, but only in one direction, and some that are complete. When the new primers anneal, These ones will only produce the size that we're looking for, specifically between. The more of those we make, the more we'll use for templates. This creates an increase in the number of individuals that are exactly the size of the primers and the space between. You can oftentimes see that there is a smear. This smear is the larger products that are produced accumulating. But the band should be the exact size expected. So let's think about how we would actually put a primer into a sequence. Let's say this is our sequence, and we want to design primers to amplify the entire gene region. In order to do this, we need to remember that DNA is reported in databases 5' to 3'. That means that we only see one sequence here. So this is our 5' to 3' strand. We need to have two primers. We need a forward primer, that's 5' to 3', and we need a reverse primer. If the strand we've been given is this one, then the strand that we can't see is its antiparallel. That means that the forward primer goes here, and the reverse primer goes here. What does that tell us about the sequence of the forward primer? Is it going to be equal to or complementary to the bases at the start? That's right, Nate. Because the forward primer is in the 5', 3' direction, it's complementary to the template strand on this side. That means the sequence is identical to the first part of the sequence in the strand we're given. This means that the starting primer, our forward primer, would look like this. What about our reverse primer? Our reverse primer is going to be in this location down here because we want to do the entire sequence. What do we need to do to this sequence in order to get the sequence for our primer? Remember that this is 5' to 3'. Is the primer sequence going to be complementary or equal to this sequence? It's a complement, that's right. So the complement is going to be this. That's an awful lot of T's. What else do we need to do to the sequence? Can we write it just like that? What else do we got to do? Nope, it's not an RNA, so we don't need to put a cap or a poly A on it. That's right, Jillian, we have to write it backwards. So remember, this is 5' on this side. We need to write our primers 5' to 3'. That means we need to flip it around. Okay, so here's our reverse primer, but that's the 3' end. So this is what we get, and we got to flip it around. Okay, so this is a reverse complement. It's complementary to the sequence that we were given, and it has to be reversed because primers are always given 5' to 3'. Any questions about that so far? So there are companies that will make primers for you. So if you have a gene you want to look at, let's say we wanted to look at this gene and we want to be able to do PCR with it. If we take these 2 primers and we send them off to the primer people, they will make us an artificial DNA primer that we can then use for all of our PCR reactions. Then it will amplify exactly what we want it to, as long as we design it right. Now, let's say instead of the whole gene region, we just want a specific area. So this time we want to design primers to amplify the first 100 base pairs. Our forward primer is going to be the same. It's part of the first 100 base pairs. But what about our reverse primer? Each of these different blocks contains 10 base pairs. So which line should we look at to find where the next primer should come from? The first line, the second line, the third line, or so on. Where's that 100th base pair going to be? The second line, that's right. So this is 61, 71, 81, 91. That means that this A right here is number 100. So if we want to include the first 100 base pairs, where does this primer need to go? Is it going to go on the left of the A or the right of the A? It's going to go on the left. The reason it has to go on the left is we want the primers to amplify only the first 100 base pairs. That means we don't want to do anything that's past this point. So the primer needs to go right here. That gives us a total size from 1 to 100. So our reverse primer is going to be located right here, with the last base of the primer at the 100, where we want it to be. What do we have to do then to this sequence to get the primer? Reverse it and complement it. Yes. That's okay, Nate. That's right. So once we know that that A is the last one we want, here's our complement, and then we flip it over. All right, so at this point, we've made 2 types of primers. We made one that amplifies the whole region, and we made one that amplifies a specific area. Now I'm going to give you a set of primers, and we're going to figure out how big the band should be. So remember, this area here for this one is 100 base pairs. That means that when we run this, it should have a band that is that size. If we see one that isn't that size, we know there's a problem. But let's say instead that somebody gave us the primers for the area we're supposed to amplify, and we don't know what size that is yet. And our primers look like this. One of these primers is going to be a sequence we can find immediately because it's the same as the 5' 3'. Is that going to be our forward or our reverse? One of these is going to be in the sequence. Don't have to do anything but find it. Is that going to be the forward primer or the reverse primer? That's right. The forward primer is already going to be in here somewhere. So here it is. There's the sequence for our forward primer. That tells us that this T is the first piece of our band product. What do we need to do to the reverse primer to get the sequence we want to look for? Find its reverse complement. That's right. So we have the reverse primer down here. This is its complement, and then we reverse it. This means that this is the sequence we're going to look for: ATG, TTG, and so forth. When we do that, we find that the primer is located right here. In order to determine the size of the band we'll make, we then identify the number for the base pairs at the beginning and the end. The first base pair is number 494. And the second base pair is number 716. So the product is 223 base pairs long. And that's what we should expect to see when we run it on the gel. Are there any questions about how PCR works, the basic steps of PCR, how to design a primer based on a sequence, or how to identify where the primers belong if you're given them instead? Okay. Once we have our PCR working, we can then use gel electrophoresis to look at the band patterns. Most of you have probably heard of gel electrophoresis before, but just as a refresher, remember that the way it works is it relies on DNA having a charge. DNA is negatively charged. The other thing it relies on is the basic rules of diffusion. Essentially, the rule of diffusion through a matrix, or in our case, a gel, is that smaller molecules diffuse faster. This means that if we use electricity to force the DNA to move through the gel, the smaller fragments will move faster than the larger ones, and we can separate the sizes of the fragments. For example, before we were looking at a 100 base pair fragment and a 223 base pair fragment. Which of these fragments would move through the gel faster? That's right, the 100. So by taking advantage of how molecules want to move already, we can separate and find the ones that we specifically want to see. We can also combine PCR with this process to do something called DNA fingerprinting. DNA fingerprinting is a way to pair DNA without sequencing. Sequencing technology is relatively recent. So to make comparisons between individuals, we had to find other ways to do that. It's mostly useful for ruling out individuals or creating likelihood estimates. And it's very quick for analyzing the identity of bacteria and viruses. The simplest way to think about fingerprinting is to think about bacteria, and specifically bacterial plasmids. Bacteria have small fragments of circular DNA in them called plasmids that contain some types of genes, usually small ones. We can also use plasmids on purpose in order to add genes to bacteria. For example, this plasmid here has a reporter gene. Reporter genes are things that make it easy to see if the plasmid is there or not. It also contains a resistance gene, resistance to neomycin. If we want to see whether or not this is inside the bacteria or identify the bacteria, we can use DNA fingerprinting processes that rely on a special type of enzyme. These enzymes are called restriction enzymes. Restriction enzymes cut DNA at specific locations. For example, the ICAR-R1 restriction enzyme cuts DNA only at this sequence. When it sees this sequence, it creates a sticky cut that creates multiple fragments. Depending on where these cuts should happen in DNA, or in this case, the plasmid, we can expect specific size bands based on the number of base pairs. And when we run those bands through a gel, we should be able to see them in a predictable way. In order to do this, we're going to separate them by size using the same process that we use to look at bands in PCR. We load the samples into the top of the gel, and then we use electrodes to force the DNA through the matrix. The DNA fragments will migrate such that the smallest fragments move the farthest, so we can predict exactly where they should be. One of the things that we need to do in order to determine if our PCR fragments or our fingerprinting fragments are the right size is to add a special thing called a ladder. A ladder is a set of known sized fragments. This gives us a reference for all of our bands. So let's make a prediction. Let's say that the region of DNA that we want to look at looks like this. And we're going to cut it with two restriction enzymes, BAMH1 and HIN3. If we know where those cuts should occur, which we do, we can make predictions about the possible band sizes we should see. For example, if only the BAMH1 sites are present, how many cuts would we expect? 1 or 2? Two. And we know roughly how big they should be as well. We know that we would have a cut at 1.8. That's going to give us a 1.8 fragment. We also know that we're going to have a cut at 3.1. That's going to give us a fragment between these two and between 3.1 and the end. So if we have 2 cuts, how many bands would we expect to see? 3. That's right. What if we only cut at hin 3? How many bands then? 2. And if we cut at all of them, how many bands then? 4. So we can use the number of bands and their size to determine which of these restriction enzyme cut sites are present. And that can be used to identify a specific type of bacteria. For example, here are the possible outcomes. And here are their sizes. By combining enzymes, we can create more complicated cuts, which allows us to create the fingerprint. So for example, we already discussed that if only hin 3 is used, or only this cut site exists, we'll see 2 bands. With both sets of cuts, we'll see 4, and with BAMH1 only, we'll see 3. This is a lot like the fingerprints on your hands. They will be consistent, and they will be unique to you. Say, for example, this is a gel that we ran. Each of these are different viruses using a lambda H3 cut, an EcoR1, CPV, BCV, and MOIV. And we want to diagnose which virus our patient has. The unknown virus, U of E. When we run a restriction enzyme digest and we send this across our gel, we get the following result. Which virus does our patient have? CPV. That's right. We can tell that they have CPV because when we cut CPV with the same enzymes, we get the same band pattern. So we can use this to diagnose and select a treatment. Are there any questions about restriction enzymes or why they might be useful for determining the identity of a piece of DNA? There are also other ways to get genes. Each of the things we've talked about so far, PCR and restriction digest, require you to know something about the sequence of the gene you're looking at, or where you expect cuts to happen. We used to stumble upon these by chance, lots and lots of trial and error. But nowadays we have other ways to get at genes, things like microarrays and gene sequencing. The only thing I'd really like for you to understand about these two processes is how they are different from each other. Microarrays select specific things that you want to look for. In this case, you're going to create little probes that are stuck to the bottom of a dish. The probes are either going to light up or not, if that sequence is present. So microarrays can allow you to tell things like if something is present, an RNA microarray might tell you if it's expressed. But what microarrays don't do is they don't give you any sequences that you didn't pick. They are not complete, and they can only give you a small part of the genome. You also have to know ahead of time what it is that you want to be able to detect. This makes them really good for quickly diagnosing things. And they're also really good for cheap sequencing. When you want to learn a lot about something, but you don't need to know everything. Actual sequencing works a different way. You don't have to know ahead of time what it is that you want to find. The way sequencing generally works is that you make multiple copies of the entire genome. Then you break the copies into parts, and you assemble the parts so that you can determine the order in which the genes occur. You can do this a few different ways. You can either compare to a reference, or you can compare overlapping parts. Because the copies are broken randomly, some of the fragments will overlap in very specific places. This allows you to put the fragments in order and reassemble the actual sequence of the original genome. When you're working on your OMIM project, when you go to RefSeq, this is a reference sequence. When your clinical variants were identified, they were compared to the reference sequence to determine what was different about them. So PCR allows us to look at specific small pieces of DNA, a specific gene, or a specific area. It also allows us to create tons of copies of the entire genome, which we can then break into pieces and use to do things like sequencing. If we want to be able to look at our specific sequences or compare them, we can use electrophoresis to do that. And we can also use restriction enzymes to cut products and see where specific sequences might be. All of these allow us to answer very basic information about what is the sequence and how is it different or similar to one we've seen before. And all of them are part of more complicated systems that we can use to treat and diagnose diseases. Now we're going to talk a little bit more about using DNA fingerprinting and why that might be helpful to us, as well as comparing things to sequences and how we can use small differences to diagnose diseases or identify individuals. Before we do that, are there any questions about the major topics we talked about today? PCR, restriction digests, gel electrophoresis, or the difference between PCR, microarrays, and sequencing? Okay, so let's talk a little bit about how this helps us and how it connects to things that you might want to do or think about for yourself. This has to do with our ideas about DNA and identity. For example, we frequently use DNA for identification. We can do this because even though most humans have nearly identical DNA, there are small differences between us that make us unique. This allows us to use DNA for a variety of reasons, but particularly to identify specific individuals or specific differences between two individuals. Let's talk about a few very specific examples, just to give you an idea. We're going to start by thinking about lactose intolerance. Lactose intolerance comes from a gene specifically on chromosome 2. And there are two genes that are generally involved. By default, humans are lactose intolerant. This is what we would call the ancestral state. We know that this is the case because all other mammals are lactose intolerant as adults. So if by default humans cannot comfortably consume lactose as an adult, why are there so many people in the US, for example, and in Europe that drink milk and eat cheese like it's no big deal? The difference is small changes in these two genes. Here are a bunch of different individuals, 6 of them, in fact, and their phenotype, whether or not they are able to consume lactose as an adult. For the most part, this sequence is identical. But there's one location where that's not true. What do you notice about the T versus C variant in connection to the phenotype over here? Which of these is the ancestral variant? i.e., the one that says that you're intolerant. That's right, it's C. This individual is homozygous for the C allele, and they are intolerant. So is this person. Is this a dominant or recessive mutation? So the mutation is not the C. The mutation is this. Remember that mutations are changes from the base state. Is the T allele dominant or recessive? That's right, Jillian. It's dominant. We know this because individuals that are heterozygous are able to tolerate milk. This tells us that the mutation, the ability to drink milk as an adult, is a dominant trait that's conferred specifically by this allele in the sequence. That's right. If the default was that tolerance was the ancestral trait, then it would be recessive. But because the ancestral trait is the C, the intolerant, it's a dominant trait. The difference between this one base change is whether or not you continue to produce a specific enzyme as an adult. In order to digest lactose, you have to produce the enzyme lactase. Lactase is generally produced by children or infants while they're able to drink milk from their mothers. And usually they stop being able to do this after they're weaned. To determine treatments for lactose intolerance, you have to determine why the lactose intolerance is happening. This is the difference between a primary and secondary phenotype. Has anybody heard of someone becoming intolerant for a temporary amount of time? Or had that happen to them? Yes, Jillian. When that person became intolerant, did they figure out why? Or did they just suddenly regain the ability to eat milk one day? They just never went back. Okay. So they were tolerant, then they became intolerant, and then they're just like, forget it, I'm not trying it again. That's fine. That's pretty common. So sometimes people will say, all right, I'm intolerant, and then later they will be tolerant again, and they'll be like, well, maybe I was mistaken. They weren't. They had secondary intolerance. This is generally a disease state. Whereas primary intolerance is inherited. So when you have bacteria in the gut can be the reason, yes, that's one reason. Or are you asking, is there bacteria? Because there always is. There's technically bacteria over here too. Ew. So the difference that happens when you don't have lactase essentially is that you can't turn lactose into glucose and galactose. Instead, the lactose stays in the gut and the bacteria that live there use it, and they use it for fermentation. That means that there's a buildup of acid and gas, and that's why it gives you indigestion. Yes, that can definitely happen. So there are lots of reasons that you can suddenly become lactose intolerant and then not become lactose intolerant. So some reasons are things like pregnancy can make people temporarily intolerant. Antibiotic use can make them temporarily intolerant. There are also stress-related conditions and autoimmune-related conditions that can do this. So there are lots of things that can cause this secondary intolerance. Now, the way that they diagnose that, primary versus secondary intolerance, is with a genetic test. So when you go to your doctor and you say, man, every time I eat ice cream, it makes my gut hurt, the doctor's going to do a genetic test on you, and they're going to see whether or not you have the protective mutation. So they're going to look at whether or not you have C, stop that, versus T. If you have C, then you do not have the protective mutation and you have primary lactose intolerance. That means you should avoid milk and cheese for the rest of your life. However, if they do the test and you have the protective mutation, that means that there is some other reason that you are temporarily unable to digest lactose for any of the reasons we just talked about. It could be because of stress or an autoimmune issue, or you need an antibiotic, or you've taken one recently. And this could result in a temporary diet change. And whenever the cause is resolved, you may be able to go back to your original diet. Or you might not, because you just don't want to risk it, like Jillian's friend. So we can use this understanding of really small differences to be able to help people understand why they might have certain phenotypes. Now, the other thing that we can do about this is we can use these small differences to be able to determine where someone is from. Did you know that this particular mutation can tell us your history? It tells us more about you than just whether or not you're able to consume milk. And it does this because of the way that this particular mutation is distributed throughout the world. So for example, here is a heat map of lactose intolerance. The areas that are in dark colors, dark blue, primarily have individuals who are not able to digest milk as adults. And the areas that are lightly colored have primarily individuals who can. What do you notice about these? That's right. That's why cheese is popular in Europe. Yes, it is hemispheric. Yes, it is based on environment. This means, though, that when you're looking at the gene, this area, you're going to find lots of C's down here and over here. And you're going to find lots of T's up here. And you're going to find a big mix in these areas. Now, part of why this is happening is because the ancestral state is C. So if we think about the movement of people, then we can see that individuals moved in this particular pattern. We're starting with C over here, and we're ending up with T over here and over here. That means that somewhere here and here, this mutation had to occur. It also means, though, that it was lost here. And we can see that by looking back at the heat map. We see movement into Europe, where we know T has to occur, across Asia and across the land bridge. T starts occurring here. And then down into South America, where we see C return. This tells us that there have been multiple times where having T develop was helpful to us, but wasn't helpful under other circumstances. So not only can we look at this and say, okay, how do we treat you for your lactose intolerance problem, or do we need to? We can also look at it and tell where your ancestors came from, and also that this trait has been helpful sometimes, but not others. For example, individuals in the USA are either of Asia or African descent if they are lactose intolerant, or they have a very rare reversion mutant, or they have secondary lactose intolerance. So if you are a person of European descent, and you know that for a fact, then you probably have lactose intolerance because you have someone of African or Asian descent somewhere far back, or you have secondary lactose intolerance, or you're unlucky enough to have this reversion. Now, we can do this a little better by looking at things like mitochondrial DNA. So mitochondria have their own DNA, looks a lot like a plasmid. Here it is, all the different genes that are on it. And mitochondrial DNA is special in that it doesn't change very much. There's a special region that changes a little bit, which is kind of funny. They call it the hypervariable segment. All they mean by that is it actually changes, whereas almost nothing else does. But it's not actually hyper. The interesting thing about mitochondrial DNA is it is not inherited the same way all the DNA we've talked about before is. Instead, it's primarily inherited from the mother only. This happens because mitochondria are passed down in the cytoplasm of the egg. So you can actually follow the matrilineage, the women in your family, based on the type of mitochondria you have. Because mutations in mitochondria are so rare, we can use PCR and genotyping to determine where those specific mutations occur in different locations across the planet. This allows us to create something called haplogroups. And this is one of the things that's used by many different companies to identify your ancestral lineage because of where the specific set of mutations you have occurred over time. This means that taking into account both your mitochondrial DNA and your genomic DNA, like your lactose intolerance status, allows you to create an extremely unique profile that's specific to you. It's also why places like Ancestry DNA and 23andMe are able to give you things like this if you choose to be involved. But there are a lot of caveats to that. For example, both of these guys here are mine. What do you notice about them? Are they the same? No, no, they are not. So I've done both of these more than once, and they are never the same. They're pretty close sometimes, but I'm actually 25% Eastern European. And I know that because my family moved here from Russia. And you'll notice here, that would be this, that's low. And over here, I don't know where it is. We got some weird stuff going on. I have no idea why that's like this. So why? Why is it like that? Well, the reason is these are microarrays. All right, so they're like, sequence your data, find out where you're from, but they're not sequencing. Instead, they are microarrays. And we talked about this idea that microarrays only detect specific things that are on the microarray. So the overlap is actually pretty bad. So let's say these are the three, I think GenCove has gone out of business at this point. These are the three big places that you can get a microRNA, a microarray done of your genome. And what you're seeing here is the similarity between them. So for example, the similarity supposedly between 23andMe is pretty high. Similarity between GenCove and the other is pretty low comparatively, and all of them only overlap by about 72%. That means that you're going to have significant differences between what these different microarrays are going to pull. So you want to go with one, you want to go with two, they're probably going to give you slightly different results, or in my case, they might give you very different results. Now, the reason for this is because different alleles that could be on these microarrays have different frequencies. And we talked about this when we discussed population genetics as well, but I'm going to show you a specific example. So this is an allele that's frequently used on microarrays to identify Native American descent. And the allele size, so that's what they're using here, the size of this allele for you, this is a repeat region, is associated with specific frequencies. So here is the frequency in the US, and here is the frequency everywhere else. And across over here, you can see the basic frequency of each of these. So 275 is this red allele, and this other one, 303, this one here, is this teal allele. What you can notice is that the frequency of these alleles is pretty variable. So if you're using this on your microarray, it doesn't tell you a whole lot about where someone is from unless they have this one specifically, 275. 275 is very specific to the America region here. And it's found in both Native Americans in North America and Native American groups in South America. So when they tell you that this percent of your genome is Native American, a lot of the time what they're relying on is how many of these very specific alleles are present. Whereas over in these other groups, this one, if you have 303, doesn't tell you anything. So you're going to get a lot more out of these types of services if you have rarer or more specific alleles. If you have very common alleles, then it will be much more difficult to tell, and you might see a lot more variability in your results if you were to do it more than one time. So it is a very fun game. And it is very useful to use things like DNA fingerprinting and these unique aspects to try and tell where we're from or try and tell medically what might be wrong. But it is something that you should sometimes take with a grain of salt because it's all based on statistics and probability. That means that it depends on how good the original analysis is. And depending on how good it is, you might get something useful or you might not. So you can have fun with these types of services, but you should be careful and taking them for face value. It's also important to do that if you have a twin or sibling you want to do it with. So you may find that you are related to somebody and they aren't, Well, that just depends on which area on the microarray they happen to use and whether or not that's going to catch that difference. So I've had siblings do this before where they didn't come back as siblings. And that is totally possible. So we think, for example, that siblings should have 50% of the same DNA, but that isn't necessarily true. They have 50% shared on average. But the range is 0 to 100%. So if you happen to have a low amount of shared DNA, you might show up on 23andMe as not being someone's biological sibling, even though you are. So that's another thing to keep in mind, right? So they're fun, and that's going to be important for all of the things that we're going to talk about next, which has to do with using DNA profiling for things like identifying criminals. Sure. Okay, so I actually can answer this question because I have interviewed there more than once for jobs and have had to write reports for them as part of that process. So basically what 23andMe does is, and this is similar to what we're going to discuss in a minute with CODIS, is that they choose areas for the microarray that tend to have big differences. So they want to pick areas that are either really inconsistent because that will give them the most variability and they could use that to figure out, what's going on, or they choose specific areas that are associated with the types of traits they want to tell you about. So one of the things that places like 23andMe are also required to do is they're required to submit things to the FDA. Right. And that's because on their microarray, they included a lot of SNPs or single nucleotide changes. So like our T to C and our C to T that we talked about before, that is an example of a SNP. They included a lot of SNPs that have theoretical connections to disease. So when they were picking the things to go on their microarray, they were like, okay, we want to pick things that we know are going to help us based on the research, determine where people are from. So we're going to pick things that have already been identified for that. We're going to pick things that theoretically are associated with health traits. We're going to pick things that are going to cover parts of the whole genome. So we might not have all of your chromosomes, but we're going to have a lot. So for example, let's say you did 23andMe. I actually have what that raw sequence looks like. 1 second. Let me go pull it. So one of the nice things about it is you can actually get your RS locations. So you guys all had to use reference sequence locations for your clinical variants, right? Well, if you do 23andMe, you're going to get reference sequence locations for all of your genome that was on that microarray. So it looks like this. So this is the full assembly, all the reference sequences, and specifically what my genotype is at that specific location. So if I want to go through and say, hey, I'm looking at this clinical variant, I wonder if that reference sequence is in the 23andMe stuff. Well, I can go look. I wonder if I'm heterozygous or homozygous. Well, I can go look. So it can be a really fun game. But again, if I were to run this again, I would get mostly the same stuff, but it's only as good as detection. So there are a few things in here. So you can see, for example, I didn't get a result for some of these. That happens too. So there's quite a few that are like that. But that's what they're picking. They're picking based off of things that they knew were going to be helpful for identifying where you're from, based on other people's research. They're also picking things that theoretically are associated with disease or other traits based on other people's research as well. And then they're picking other SNPs where there's high variability, but they don't know what it causes because they're banking on researchers finding information about those SNPs. And when they do, they can make you pay for a new report about what that might mean for you. But that's why. Does that help, Jillian? Yes. Yes. And of course, we've also talked about this idea too, that genetics is not fate for a lot of things. So just because you have a SNP or a specific sequence that 23andMe or Ancestry or even your doctor might be looking for, it doesn't always guarantee that you have whatever that trait is. And so that's one of the reasons why the FDA is really involved in regulating what they can and can't tell you. So, for example, I have my raw microarray data. I can go through and I can look for disease variants if I want to. But only the ones that have been approved by the FDA can actually be given to people on the platform. And that's because it's medical advice and that is highly regulated. So that goes into some of that ethics stuff too. Like, what do we do with that information? When before that happened, before the FDA got involved, for example, 23andMe did tell people about their BRCA status. So remember, BRCA is a mutation that causes breast cancer, but they didn't provide any support for people who had the mutation. And so people would say, oh my God, I have a BRCA mutation, I have breast cancer, and that is not how it works. And so the FDA got involved, they made them pull all of that, and now they have to provide genetic counseling if you choose to look at those variants, right? So there's a lot of stuff in there about that. So you guys will be in a much better place should you choose to do this than the average person. No, got that. All right. So all of this connects back to this idea of genetic fingerprinting and profiling. And one of the things that we tend to think of using it for are forensics. So lots of people who are interested in biology, interested in being a doctor, interested in being all kinds of things like crime shows or dramas or stuff like that. And of course, we do use this process as well to identify things from crimes. This is usually referred to as DNA profiling, which is just a type of DNA fingerprinting. They just think that I think profiling sounds cooler, so that's what they like to call it. This type of profiling or fingerprinting is used for paternity tests. It's also used for crime scenes. It's used to identify missing people or corpses. And we also use it in the military, for example. So here's a long list. So we use it for forensic cases, paternity tests, historical investigations, try to determine ancestry, when we're trying to figure out immigration status or other issues for people who won't tell us where they're from, when we're trying to find missing people. We also use it as a military dog tag. So a lot of people in the military will have their DNA archived. So if they are found, they can be identified. And we use it to identify or manage populations when it comes to things like ecology and conservation, because we can use this to track where specific organisms are coming from or where they're living. Most of the time, this relies on something called microsatellites. So it's a little bit different from the restriction enzyme version. So microsatellites are short tandem repeats, and we talked about what a short tandem repeat is and how they are variable. So for example, Huntington's is an example of a disease caused by this. Huntington's disease happens when you have too many copies of the STR. So more is not always better than less. Most microsatellites do not cause disease, and instead they're just variable lengths between individuals. So you can use those different lengths to identify someone. We detect these by PCR generally, and then the fragments are represented as peaks on a graph to tell us whether or not the person is homozygous or heterozygous. And then we can compare those peaks to determine if the people are the same person. Most of these different regions have multiple alleles. So we've talked about the idea that while we do our genetic problems, we're usually assuming that there were 2 alleles, but the reality is that there are generally hundreds of alleles for most regions. So for example, here we're looking specifically at the number of copies at this specific region. And you can see that there are quite a few different copy numbers that are present here. These kinds of tandem repeats are spread throughout the genome. So for example, this is just on the Y chromosome. And all of these are locations where you could find an STR repeat that you could use to identify this person. Right, and that's one chromosome. Most of the time when we talk about this from a crime standpoint, we're referring to CODIS. So CODIS is the database of STR loci that are used by the FBI. And here they are. These are the primary CODIS regions that are used, the number of alleles for each of these regions, and the number of repeats, the chromosome they're on, all of that. All right, so you can see that there are some alleles that have a lot of variability, like this one, and there are some alleles that have less, like this one. And this has to do with the number of repeats, so how many you might find. Generally, a CODIS match or an identification match is based on how many of these are going to match at the same time. So for example, if we were to look at these as a picture, these are the general distribution of the primary 13 CODIS loci. So a full match would be a match at all 13 locations. The likelihood that you would match at all 13 locations is very small, but it's not zero. And the reason for that is because of the high allele number. So for example, VWA has 26 different alleles, whereas TPOXX has 17. And each of these alleles has a different frequency. So to give you a specific example, the chance of two unrelated individuals matching at 9 to 10 loci would be 2 in 100 million if they were all the same race. So the more places you match, the higher the likelihood. Right, so two individuals in 100 million would be expected to match maybe 9 to 10 pairs, but not all 13. When you start getting up to a 13-point match, you're talking about relatives, close relatives would be the only other option. All right, so this is what CODIS is actually doing. It's looking at STR allele lengths, and then it's making comparisons between those lengths from two different sources based on the rate of that allele within the population. This also means that the likelihood of matching is going to be different based on the population you're pulling from. So the likelihood where you live specifically that someone would have the same match to you is the one that's actually relevant. Of course, we look at these matches also by running them on a gel. So for example, we could look at these two individuals here, and we could see that their STR locus is not the same at this specific site. We do this by running PCR with standard primers, and the only difference is going to be the length based on the number of repeats. So the more repeats, the larger the band. So that's how we're telling them apart. We're not sequencing them. We're not making sure that all of the different, amino acid base pairs are in the same place. We're using size. This also makes it cheaper because you don't have to sequence them specifically. We can also tell whether or not the person is homozygous or heterozygous for the repeats that they carry. If they're homozygous, then both of them will be the same length. And if they're heterozygous, then they will be different lengths. And we can see this by looking at the pattern on the gel or in the read bands. So for example, down here, we can see that this person has a peak at 22 and 24. This person has 23, 24, and this person has 21, 25. If we were to look at multiple places on the same chromosome, we could also tell when and where this person is homozygous. So for example, these tall peaks are a person who is homozygous, and these shorter double peaks are heterozygous locations. When you're looking at paternity testing, this is particularly important because only half of the peaks will be given to an offspring. So you need to know all of the possible combinations. So when we're making a CODIS match, we might have something that looks like this. We'll have a CODIS match of peaks from the crime scene or location that we want to compare, and then we'll have match peaks from people who could have been or were in the location at the time. We can then compare what those peaks look like and determine which of the individuals we cannot rule out. So based on this one, would you pick suspect 1 or suspect 2? 2, that's right. Suspect 2 has peaks at all of the appropriate locations. It's important to remember that CODIS is not proof of guilt. It can only be used to rule individuals out or not. Because there is an extremely small likelihood that a random person could have the same matches, especially because getting all 13 to work at the same time doesn't always work out. It's mostly used to prove whether or not a person has lied or has been in a location before or not been in a location before. Nowadays, we can run multiple CODIS hits at the same time. This creates a really pretty looking thing that looks like this, right? 2 different individuals run at the same time. Same idea. One of the things that's also often used are standard peaks. You'll notice the standard peaks are gold here occur at all locations. This is the same thing that a ladder would do in a regular gel. You can use this to determine where the peaks are in reference to the standard, because sometimes things don't run exactly the same way, and it lets you standardize. CODIS is a powerful tool, and it has been used to convict people based on only their DNA. But that has some controversy. An example is this individual here. Cold case files from back in the 80s and 90s eventually identified this particular individual using a CODIS hit. The SCR profile did not perfectly match, but they did match each other, which told police that the same person was committing all of the crimes. This is what running this actually looks like. So here is an individual who is running a CODIS fluorescence hit to try and get a match. Now in CODIS, we're looking for perfect matches, but in paternity testing, it's half matches. So for example, if we were to look at these individuals here, we can either exclude or include an individual. The first father here cannot be the father. The reason for that is in order for the child to inherit a 5 and a 3, he has to get 1 from each parent. He inherited the 5 from the mother, which means the 3 must have come from the father. Since the father does not have a 3, he can't be the parent. The second father cannot be ruled out. He does have a 3 that could be provided, but that only makes him a possible father. It does not mean that for sure he is the father. To determine that, they usually calculate a special thing called CPI, which you probably will never have to worry about, which I hope you'll never have to worry about. It's based on the frequency of alleles at multiple locations. If the CPI is 0, which it is in this case, at the top, then there's 0 chance that the person could be the father. But in this example, we have multiple loci that are being compared at once. We also have the frequency that these loci appear together. 12, 11, 21.2, and 15 would have to all appear together in order for them to be possible to be given to a child together. This creates a paternity likelihood that is much higher because of the multiple loci, just like the multiple loci in the CODIS hit. We use the same process to identify relatives for individuals that are lost. For example, here is one that came from a 9/11 victim that was identified from remains. So when victims are found, we can frequently tie them to their living relatives by looking for the alleles that they have in common. We can also use this to determine whether or not individuals in embryos to be pre-implanted contain alleles we do not want them to contain. Say, for example, that one parent has an allele that is deleterious or bad. You can screen embryos to look for whether or not they contain the signature of that allele in the same way. This is really common for in vitro fertilization to avoid lethal combinations. And we can use the same practice to try and do things like find ivory or deal with conservation efforts. And one of your activities will be based on this. Where did the elephants come from? Where did the ivory come from? And how did it get there? So hopefully today I've helped you understand at least a little bit about the most basic concepts and techniques that are used for all of the different things we're doing with genetics and biotech. Yes, yes, yes. We do this a lot. But I also hope I've helped you think about this idea of should we do this? Doing things like PCR, using gel electrophoresis, getting sequencing, and using that to identify individuals can sometimes come with problems. Does a person who's diagnosed with a genetic disease need to tell people related to them or their partners? Should the government be able to use things like DNA information from Ancestry or 23andMe to solve crimes, especially given what you know about how accurate they might be? And should our DNA profiles be used as identifying features, like our social security numbers, are all problems that come up. But there are positive things too, like being able to explain why certain traits are being passed on, or whether or not something is a primary or secondary disease. Now, next week, we'll talk a little bit more about some detailed things, like how do we make changes to actually identify what's happening, using things like CRISPR and RNAi. And we'll also talk about using GWAS studies to identify traits when there are multiple genes involved. I'm sorry I ran a little bit over today and that I started a little bit late, but hopefully it was interesting at least. Are there any questions before I let you guys go? All right, if you don't have any questions, I hope you have a wonderful Thanksgiving. Yes?
I just have a question, but it's kind of more like related to my project. So I don't know if anyone else cares, but I don't know if anyone else has like more general questions before I get into it.
All right, I'm gonna stop our recording just in case.